be going on for the rest of the term is figuring out basically how to model and understand cell growth in bioreactors. So the first part of understanding cell growth is understanding what happens in a batch situation. So how do cells grow when you stick a small number, which is called an inoculum, of cells into some kind, it could be a flask, I mean anything really counts as a bioreactor, it could be a plate, a flask, something where you have limited amounts of medium and you run through it. So last time we talked about how you actually measure that number of cells. And we talked about a number of different ways that you can do counts or dry or wet mass, cell pellet volume, optical density, use a Coulter counter, use a hemocytometer, all kinds of different ways that you could count the number of cells as you're going through to understand what's happening in your process. So let's say we use one of those things and we look at what happens over time. And it could be cell number or mass or whatever you have. What happens, so you've got a bioreactor, and I know some of you have now done this for your lab course. You have a bioreactor, you have a flask, and you put a small amount of your cell of interest, whether it's yeast or E. coli or whatever you want, in there. What's the first thing that happens? You've got lots and lots of medium, lots of nutrients. The first thing that happens is rapid growth. Actually, no.
Why isn't it a lag period version then?
Right. Right, so you have some kind of lag period, which actually potentially is reasonably significant. And for those of you who have done the yeast bioreactor and for L2, it's actually quite significant. So it takes a long time for those yeast to get rapidly growing. And we had lots of, I heard lots of complaints last year, not my course, but I still heard them last year from people who ran through it because initially the students themselves were doing the original inoculation, which meant that pretty much the whole lab period was spent during the lag phase. So they got no meaningful data. Now that gets inoculated for you in the morning, which is a big difference. So lag phase is pretty significant. So we've got lag. Why do you have a lag phase? Why does it take a while for the cells to start growing?
When these species are probably kept dormant before they put into the bioreactor, so usually they're kept on the cold reactor form, so they don't allow them to wake up.
So for the yeast, you've got a few things going on because they've actually been freeze-dried. So they have to become hydrated and then become metabolically active before they even start using nutrients. So that's a particularly harsh thing to do. But say you had even, and this is actually something we've been trying to talk to 4L2 people into doing, say you had even, so what you do, say when you start bread, you take your yeast and you let it sit for a little while until it's rehydrated with a little bit of warm water and sugar, and prior to adding it to your dough, you have it sit there for a while, and the reason you have it sit there for a while is to get it into the point where it can be metabolically active, is rehydrated, and is ready to go, and then once it's sat there on your countertop for a little bit, then you put it into your mixture of flour and water and sugar and all that makes your bread, and you use the carbon dioxide product to make your bread all puffy. But even in that case, there is a lag phase. So even when you have metabolically active yeast going into a nice, yummy area where there's lots to eat, you have a lag phase. So actually, if you were to, say, measure, when bread is rising, you put it in a nice warm area so that the yeast can be at its optimum temperature. And it takes a while for anything at all to happen. And then it starts to happen quite quickly. So why would it still take a while for anything to happen, even if you start with a metabolically active inoculum?
I guess it takes a while for the nutrients to be uptaken.
Kind of, but even if so there's two possibilities. One is that you've got your inoculum in different conditions than in your larger reactor, and that's definitely the case. So if you start growing it on lactose and move it to a reactor where it has to grow on glucose, then it has to get different metabolic pathways up and running so that it can adapt to its new nutrients. So that definitely adds a pretty significant lag phase to it if you've got a different growth medium in your inoculum as you have in your large reactor. And that makes sense, right? You have to get new enzymes expressed and upregulated, active. So you may actually need some gene expression to occur, followed by protein production before it can adapt to its new circumstances. So that's a pretty big contributor to lag phase. Can you think of anything else? What happens when you take your small inoculum and put it into a big bioreactor? Yeah, so even if it's the same, make sure you pull it close. Even if it's the same nutrients, suddenly it's become incredibly diluted. So actually most bacteria are really good at quorum sensing, which means telling how dense the population is around them, and they respond to signals from the other cells around them. Mammalian cells do the same thing. I'm less familiar with yeast biology, but I expect it also does the same thing. Even at minimum, by sensing how much nutrients there are left versus how much metabolites from the other cells there are available. So just, even though it seems like it's the same situation, it actually isn't, because you've got a different nutrient concentration, because if you've grown your inoculum up to a fairly dense, or a fairly high cell density, you've used up a lot of your medium, or a lot of your nutrients, and suddenly you're putting it into a very dilute circumstance where you've got lots of nutrients and no metabolites that it can really sense. So all of these things contribute to the lag phase. In fact, even if there are some circumstances where your glucose or your lactose or whatever your carbohydrate source might be your limiting nutrient, but if you have it where there are other micronutrients that are not available or not as available in your nutrient broth, although those things might not eventually limit growth, it takes longer for the cells to adapt to be able to compensate for the missing micronutrients. So you can make things easy for cells and give them proteins in ways that they already want them, like growth factors, or you can make it more difficult for cells. So they're capable of synthesizing those things themselves, but it takes a lot of metabolic energy to do so. So they spend their first while basically adapting to whatever the new environment is. So people did studies, for example, where they put in different amounts of an enzyme cofactor, magnesium, and if there were very small amounts of the enzyme cofactor there, then the cells could survive, but they had quite a long lag time, whereas if you put in copious amounts of the enzyme cofactor, then it decreased the lag time significantly. We don't spend a lot of time modeling lag time. Not a huge amount is understood about lag time and how to deal with it mathematically, but you can imagine if you've decided that the way that you're growing up your product is in batch reactions, if you're spending a quarter to a half of your time in lag time, that's a significant cost to your operation, right? Because it takes a long time to get each batch through your system. Yeah?
So are there any empirical models that can estimate how long their lifetimes will be?
Not as far as I know. So there are a lot of rules of thumb to decrease lag time. Generally don't switch nutrient broths. Another rule of thumb for keeping a relatively low lag time is only diluting 1 in 10. So it's actually, most cells have a very difficult time adapting to a dilution of more than 1 in 10. So in some ways, like what that means, so in 4LL3, you have these 1 or 2 liter bioreactors. There, what we do to make sure that the lag time isn't too ridiculous is you actually have a very small inoculum that you that maybe has been frozen at -80. That gets streaked onto a plate so that they're active, and then grown in a flask overnight. So you actually get a reasonably large sized inoculum, about 1/10 of your reactor, that you can put into the bioreactor of actively growing happy cells that are in the same medium, same nutrient medium as your bioreactor. So what that means, sort of from a production point of view, is if you're looking at very, very large bioreactors, you may want actually a series of smaller reactors to essentially grow up your inoculum. Because otherwise, you think about it, I mean, never mind lag time, but you think about how effectively you're using your space. And if you have a bioreactor that's the size of this room, and you put in an incredibly small inoculum, that inoculum would have happily grown in this size, and then in this size, and then in that size. So you're not really using your resources effectively, never mind the lag time, which would be incredibly significant. You're not using your resources effectively if you're using your incredibly large bioreactor to grow 1,000 cells, and then 10,000 cells, and then 1 million cells, when really what it wants to grow is 1 trillion cells, and 3 trillion cells, and 10 trillion cells. Right? Makes sense? So the lag time is something that tends not to be discussed a lot. It's very difficult to model. It's difficult to predict except empirically. The other thing, of course, you're moving it from a plate where, say, they're growing basically on a surface to growing in suspension in a stirred flask. Well, that's a different environment. So they have to suddenly adapt to not growing on a surface with air around it, but in a stirred flask. And then they suddenly have to adapt to a larger reactor that has air being sparged through it and a more significant shear stress acting on it. And although it seems like similar conditions, it may in fact be quite different conditions from the perception of the cell. So lag time does tend to be reflected empirically, but it's if you're in a plant and using batch reactions, it's actually a pretty serious thing. So just let's talk about batch reactions for a second. So a lot of biological reactions are done in a batch format. And there are good things and bad things to doing things in a batch format. What are some good things about doing things in a batch format?
It's easier to do like if something goes wrong, it's easier to contain what goes wrong to one process. Like in a continuous process, if something goes wrong downstream and you don't catch it in time, you ruin a lot more potentially than just one batch going bad.
Yeah, so from a risk assessment point of view, if you can monitor a batch and if it goes bad, you can just check out the batch. Whereas if you have one CSTR that's running continuously and something goes wrong in the process, you've have to get rid of a lot of your product, and B, have your entire process go offline. So if you have a number of small batches, it's much more resilient to things going wrong. So that's one bonus. What's another reason that pharmaceutical companies in particular might gravitate toward batch reactions? It's not a scientific reason. Well, not really.
Quality control.
So kind of. So one piece of background here is that's actually no longer the case entirely. because you can do very effective quality control by monitoring different aspects of the process as they're going on. But having said that, yeah.
Would it be easier, like, particularly pharmaceuticals as well as like large organic molecules are really hard to synthesize, so would it just be easier to synthesize something in a batch process when it's really complicated and has many steps?
Potentially, and that's actually a really good point because of how some of these things are synthesized or made, and we'll talk about that later. But that is a really important thing, is there are some ways that they're made that are best adapted to a batch process. But it's partly because of points of view and the way people have been brought up in a lab environment who have been the people who primarily work in pharmaceutical companies. And then as a consequence of that, some of the regulations that used to be in place for how to run the pharmaceutical companies. So typically when a drug is made, it gets made sort of like the penicillin story. It gets made in small quantities in the lab, and it's a batch process that makes them, and then they look to scale it up, but those are still batch processes that make them, and then they, after a certain point of period of time, the scale up is because it becomes that you have a number of batch reactors that you can put into service that are running sort of at the same time. And that's your scale up. Now, the FDA and in Parallel Health Canada for a long time felt that the quality of the product was dependent on how the processing went. So if you were going to make a process or make a product in a batch reaction, their fear was that you would come out with a different product with different contaminants that needed to be purified from it in a continuous process. So if all of your and you're not going to put a fancy continuous process in to make small quantities of stuff for a clinical trial. But if all of your clinical trial work was based on a batch reactor, then you had to continue making your product in a batch reactor from there on out, because only the product made in the batch reactor was approved. And if you made it some different way, then that might be a different product that you'd have to go through a separate multi-million-dollar clinical trial to do. Attitudes have changed, and I think about 15 years ago, they did change the way that they regulate processes in pharmaceutical companies. So now it's much more accepted that you can have these more chemical engineering-like processes grow up your bacteria or other biologicals, and it's okay now to show that at every step of the way you have appropriate quality, and that'll lead to an appropriate outcome at the end. Whereas in batch reactions, you tend to kind of stick everything in, hope for the best, come back at the end, test it, and see how it went. As a result of these changes, I think we've actually seen a lot more engineers come on board, at least as consultants, setting up processes in pharmaceutical companies, because there's a much higher acceptance of fancy production methods like continuous methods. But having said that, not all biologicals are open to that. Another downside of batch reactions is that you have to be very, very careful about cleaning. So sterilization becomes incredibly important. You have the flexibility that maybe there's a big demand for drug A, you can put all of your batches into production of drug A. Maybe for flu season, for vaccines, you can move all of your production over to one thing, and then you maybe want to make a big pile of something else. So you have a lot more flexibility in your process if you use batches. But actually, a big percent of the time in doing batch reactions is spent emptying it out, cleaning it, sterilizing it, making sure all of your toxic sterilizer is washed out with sterile water, which is in itself expensive, starting your process up, heating it up, getting all your parameters appropriate so that you can then inoculate it again. And there are possibilities for error at every step of the way. So although it might seem safer, you don't lose as much, there's a higher risk of something going wrong each time. Because maybe you weren't careful when you grew up inoculum B, and this actually happened in 4LL3 last year. The students only grew up, so take this as a suggestion. So one of the things you have to do is grow up your tube of, you pick a tube of inoculum and you put it into a shaker flask for overnight. Do a little bit extra, a little bit more than you think you need to, because there's always a small risk that something that you do along the way with your tube of inoculum the next morning might lead to something that doesn't have the cells in it that you think. And then what are you gonna do? You're stuck. You don't have anything, any backup. So always have a backup. That's just my suggestion for 4LL3. But it's a serious process consideration if you're doing batch reactions, because these are the sort of process considerations you have to think about, that things can go wrong along the way and your scheduling can be all messed up if you don't have backup plans. Okay, so that's my little spiel, and I'll talk again about some of the advantages and disadvantages biologically of doing batch reactions as we start talking about how to actually make products. So we've got lag time, and then what happens after lag time?
Log phase.
Log phase, so what does that mean?
So it's like exponential growth.
Yeah. Yeah, so this is what we were talking about briefly last time, where your growth rate is dependent on how many cells there are. So you have a constant specific growth rate, which means that if you have twice the number of cells, you have twice the growth rate, and everything doubles and doubles and doubles and doubles, because that's the way cells grow. I was actually explaining to my so my son is 4, and to get him to brush his teeth, we talk about how the sugar bugs are going to eat his teeth. And he started asking how sugar bugs have babies. So I had to try and explain that, in fact, they just double. They eat a lot and they get bigger and bigger, and then they just double. But
Well, that's how humans do it too, right?
That makes very simple explanation.
yes.
So long, kids, so close.
That's where all that food's going to. Suddenly, there's going to be a quivering Carl in the front of the class. No, there will be 2. But yes, so you have doubling and doubling and doubling and doubling. But if we're talking a batch process, then what happens?
It'll like taper off.
So it tapers.
And eventually it just reaches like a plateau for a while.
Okay, plateau. So why does it have this plateau? So let's just log phase or exponential. So this is happening when you have more than enough nutrients. There's no limitation in nutrients.
Yeah, the plateau phase will be happening because there's not enough nutrients to sustain like a constant population. There's not a limiting amount of I think that the nutrients and the amount of yeast or whatever are limiting each other basically. Like there's not so few nutrients that they're starting to die off. They can't all live, but there's not so many nutrients that there's enough for everyone to just have like.
So yes, essentially. So you start being, there are actually a couple of reasons you can get to this point. With yeast, there's more than one reason. So if you have well, first of all, you can have substrate limitations where you can actually be poisoned by having way too much glucose at the start. And we'll talk about that and how to model that. But say you actually have a lot of sugar in your fermentation medium. So, for example, you grow grapes that have incredibly high sugar content because you had a really hot fall, and they ripen really well, and you make them into your grape juice, and you go to ferment them into wine. There probably are still quite a few residual sugars left when the yeast stop growing. So in that case, they haven't run out of food. What's happened?
Be a byproduct of the producers right now.
Such as, yes, exactly. So yeah, yeast typically can only grow up to like 8 or 10% alcohol before their product starts poisoning them. Basically, they drown in their own pee.
That's what wine.
Is it?
I thought that was.
So that's what.
15 wine.
Yeah.
I thought
So
that
it's,
the-
yeah,
that was wine.
you're right, it's about, it depends on the strain of yeast. So wine by itself is, yeah, you're right, it's
Is
about,
that what they do, just go until it's,
basically until it gets as much alcohol as you
Out
possibly
of
can.
curiosity.
But there are different strains that have different resilience to
Is that
alcohol.
what beer does as well?
To some extent. So to some extent, it tends to be more limited by time. You also don't have as much sugar typically in a mash available to be converted into alcohol. There, your other byproduct that can limit growth is carbon dioxide, which is why it explodes. So it can end up being limited by some of the, yeah, and that's why if you want something higher, you have to do things like distill and that sort of thing. Actually, with beer, again, they actually, at least the big brewers tend to make it to a higher amount of alcohol than they actually want and then dilute it down. Because it can be hard to control getting to exactly the right percent alcohol. And with the small brewers, you're never quite sure that it's the 6 or 8 or 10% that they say.
I think they have like a 1.5% tolerance, but then.
Yeah, they do, and they do test.
Yeah.
And the LCBO will not take it if they don't pass those tests, but there is tolerance. Whereas the big brewers actually are have very consistent quality control, but maybe get to a less flavorful result. Okay, so stationary phase, you might be starting to be limited by something you've made. Probably you're starting to be limited because you've run out of nutrients. Now, a couple of things can happen when you run out of nutrients. You think about what happens to animals. You run out of nutrients. You don't just die off immediately, but you stop having babies typically. So that's one thing initially in the stationary phases. The cells aren't so out of nutrients that they die, but instead of putting their energy into making more of themselves, they're putting their energy into metabolic maintenance. So for a while, they just redirect their energies into staying alive rather than making more, because they recognize that their substrates are becoming limiting. When they become a little bit more limiting and they start starving, one thing that can actually happen is that the cells will catabolize one another. So some of the cells, they cannibalize each other. Some of the cells start dying and they release nutrients, and those released nutrients from the dead cells actually can be used by the remaining cells. So you can actually, sometimes it's not quite a stationary phase, it'll start to go down and then back up again, which is called cryptic growth, when they start using nutrients from dead cells. But basically what you're seeing here is they're stopping from reproducing as much, and or the death phase is equaling the division. And then after we get past the stationary phase, then you go into your death phase, where they really are starved of nutrients. They really don't even have enough left to stay alive, and they start dying off. So obviously, you're gonna wanna start, well, for the most part, if you have a batch reaction, you'll probably wanna harvest it before it gets well into the death phase. And we'll talk talk about this a little bit more in a second.
What's the general like scale on that, on those 3 staging E's?
So, let's say this is actually, if we do this as, if I do it instead of that, if I do it more as a straight line, and I do it as a log cell number, this is variable. It's usually actually shorter than this, so it's pretty short. But it's not unreasonable to have a timeline on the log phases sort of similar to on the stationary phase. So especially yeast cells and bacterial cells, they're actually remarkably resilient. It's hard to kill them. Even some mammalian cells, it can be hard to entirely kill. So, so remember I told the story about the guy who totally messed up his 4th year project by not doing his early checks? Same guy, over the Christmas holidays, he was growing up CHO cells, which are Chinese hamster ovary cells. They're sort of if you have to make something in mammalian cells, that's the workhorse cell that you choose. Those are the cells that really have been optimized to effectively make proteins. If you have to make products in mammalian cells, typically it will be in CHO cells. So a lot of the optimization work has been done with transfected CHO cells that make lots of stuff. And that's partly because they really grow, as far as mammalian cells grow, they grow really well. So typically what you're supposed to do to keep mammalian cells happy, they like to be on surfaces. Although CHO cells actually can grow in suspension, that's one of the benefits of CHO cells. But once a week, you remove them from the surface with trypsin, and you take a small dilution, usually 1 in 10 dilution, start to seed it onto a fresh plate. 4 days later, you take the medium away and give them fresh medium, so that you feed them again. And then, 3 days after that, you take them off the surface and dilute them and keep them going. Well, he was gone for almost a month over the Christmas holidays, and he just left them. And typically, either you get somebody to take care of them if you're not going to be in, or you freeze them down so that they're not doing anything at all during that time. And when he came back, he managed to resurrect them from the incubator dead and kept them going. Now, I do not recommend that, because that's the sort of thing that selects for really odd behavior in your cells. It can cause a lot of genetic mutations, and don't do it. But these cells, like as far as they go, these cells can put up with a lot. And that was it. They were probably about there in their death phase, and they managed to come back. So you can do it. I don't recommend it. Yeah.
Do CHO cells need like the supporter cells that cells need?
No. So mostly, really, most of the only cells that need support cells is embryonic stem cells. Almost every other immortalized cell line will grow on its own. And yeah, CHO cells will not only grow on their own, they'll grow in suspension if you need them to. So they're pretty sturdy cells as far as mammalian cells go. So I actually, during my PhD, I did some transplantation studies with CHO cells, and I needed to do an actual graft. So I got some actual Chinese hamsters in. Cutest thing. God, they have these big eyes, and they're really fuzzy. Little tails. So I don't recommend doing it.
What did you do after?
Well, they die.
They die or did you kill them?
Yes.
Yes.
Well, they die because I killed them. But they're really cute if you go Google pictures of Chinese hamsters. And so the normal ones you get at the pet store are the Armenian hamsters, and they're actually really mean. Like they bite you, and they're nasty, and they're big and not that cute, and they're pretty smelly. Chinese hamsters are really cute and adorable. a pet, I recommend Chinese hamsters. But yeah, maybe don't do experiments on them. But I mean, yeast cells actually can like not necessarily die; they can just go dormant completely. E. coli typically will start to die, but is still pretty resilient. Like it's hard to kill bacteria, so that stationary phase can be pretty long. They can just almost go into stasis and go into really like needing quite a low metabolic load. So it can be quite long. And one question is, why might we care about that? And we care about it because sometimes what we're making gets made during the stationary phase. So if we're genetically engineering organisms, then typically it'll get made during the exponential growth phase. And it'll be what's called the growth-associated protein, which we'll talk about in a minute. But if you're looking for something that's made more naturally, sometimes those are what's called secondary metabolites that get induced when they're in the stationary phase. So maybe when they're under some kind of stress or pressure is when secondary metabolites get made. So for example, if you think about secondary metabolites from plants, a lot of them only get made when the plant is under stress. So drought stress or nutrient stress, something like that. So if you're making some hormones, some things that the cells will make naturally, you'll see quite often that they actually get made during the stationary phase, and you'll have to go through the growth phase separately, and then during the stationary phase is when you get the actual product production. So it's important to know about the product that you're making to know which part of that is most important. And you can see that it would be difficult actually to get yourself into a happy stationary phase in a CSTR type reaction. Because here you're not supplying fresh nutrients to your cells, they're not actually dividing. So if you were constantly streaming things through your reactor, you would just be washing out your cells and they dilute to nothing, or they revert back to this exponential phase if you're putting the nutrients in. So there are some, so this is sort of what you were saying about biology, there are some cells that you actually need, or some reactions that you actually need to do in a batch situation because you need to get them into that stationary phase.
CSTRs don't make the stationary?
Well, if you think about what's happening in a CSTR, typically what's happening is you're putting growth medium in at a constant rate, and there's cells being actively dividing within the CSTR. You keep that cell concentration constant and dilute them out by
So you can't get secondary metabolites from CSTRs?
Not easily, not easily. You might be able to come up with some sort of recycle mechanism, but then you're essentially just reproducing the end of a batch reaction. So typically you would not use CSTRs for secondary metabolites. Typically you would have to use them for batch reactions. Okay, so let's put a little bit of math to this. We kind of did this before. So in the exponential phase, the exponential phase is the part where it's actually easiest to do your math. The log phase is really hard to, or the lag phase is really hard to predict the time, but it's really easy to predict your cell growth because it's 0. Stationary phase, similar sort of thing, but again, your cell growth is 0, so it doesn't really need modeling in any way. Death phase, we can, well, it does, we can start to do a little bit of modeling in the stationary and death phase. But predicting when those transitions happen can be potentially challenging, and we'll talk about ways we can do that. So in the exponential phase, we said so X is our cell concentration. So our rate of cell growth is proportional to the number of cells we have, and the proportionality constant is our net growth rate, our net specific growth rate. So that's our first order exponential growth rate expression. And that's our specific net growth rate. Okay, so easy, we bring that x over, so dx divided by x equals mu net dt, and you integrate and you get it's not difficult integrations typically. Or you can predict, you want to predict how many cells you get at a given time. You say it's the amount that you seeded it with times mu net times time. Okay. So if I give you, if I say at the start of exponential growth phase, we have a concentration of X, how many cells do we have 2 hours later, then you can figure that out if you know your net specific growth rate for a specific growth condition for a specific cell. Okay, make sense? Okay. Similarly, so if we then plot 1x versus t, so you count your number of cells at different times. We plot ln x versus t. Your net specific growth rate is just your slope, right? Okay. So how long does it take to double? So we want to know what our doubling time is.
X2 equals 2x1. Or sorry,
Yeah, x
whatever.
equals 2x naught. So x equals 2x naught. So I think Ryan added, you could say ln 2.
Yeah.
Okay. So now we've got ln 2 equals mu net t, or we can call it t doubling time. Tau d is doubling time, which is just the t that you get when you put that 2 in there. Okay. Equals ln 2 over mu net. Okay. Or 0.693 over mu net. Okay. You can do a similar thing if you, so it merges back and forth between whether people are looking at cell number or cell concentration, but it comes out about the same. Okay. So that's pretty easy, exponential growth phase. Now, so this is the other thing I didn't actually really mention, that's just called the deceleration phase. Okay. And that's, the deceleration phase is again where they're just adapting to a new, less happy environment. So they're still dividing, but they're spending a lot more of their metabolic energy changing into a cell that'll be happy with low amounts of nutrients. Yeah.
Sorry if you already answered this, but is the cell death phase just the negative rate of the deceleration phase?
So close, actually really close. So the concept is very similar. So in the stationary phase, we've got something called maintenance energy, which we talked about briefly. So that's, now we've got, instead of all of your energy going into dividing your cells, or almost all of your energy going into dividing your cells, you've got some of your energy going into just keeping your cells alive. And at the stationary phase, that's an increasingly large part of it. So we model that by saying dx by dt equals minus kd x, or x equals x s naught e to the minus stationary phase, what you're probably actually having to get it truly stationary is a balance between your cell growth rate at that point and your cell death rate. But really what this is saying is this is actually, you can think of it more as the energy that's going into maintaining the cells. So you've got a much decreased cell growth rate at the stationary phase. And then this isn't really a cell death rate. This is a cell maintenance rate. So it's like saying we're still using nutrients, only these nutrients are just being used up to keep everything the same. Or in other words, if you run out of nutrients now, then those cells are going to start disappearing.
What's the XSO again?
At the start of the stationary phase.
Okay.
So X naught is always saying your initial period, and the reason it's SO is because it's
Station.
the start of the stationary phase. Because obviously, so this is going to be your X naught there, right? So you can't, so here would be your S naught. Okay, so basically you have to completely change models once you get into different phases. Okay, and then death phase very quickly is the same. Okay, here we actually, so I'm going to switch from concentration to cell number because it's in the death phase, it can look like your cell concentration is the same because you have all of these cell debris that are affecting your optical density, still will fill your cell pellet space. So it's not always really accurate to be based on concentration, but your number does in fact go down. Equals minus KD prime n, or n equals ns e to the minus KD prime t. Okay, so that's the start of the end of the stationary phase. The number at the end of the stationary phase is ns. Okay, and there if you do 1 n versus t, you've got a straight line with a slope of minus KD. Or actually KD prime. It's KD prime because it's looking at cell number and the actual death rate as opposed to the maintenance rate. Okay. So those are our basic models. N is number of cells, X is concentration of cells. And the reason that we switched is because it looks like the concentration may affect